Welcome everyone, guests, panelists, to this series that we call Compelling Conversations. Compelling Conversations is an exclusive virtual opportunity for partners, alumni, and supporters of the School of Physical Sciences to participate. These are live events, they're unscripted, and they're conversations among world experts on imperative topics. This is our fourth installment, and we'll follow it with a fifth in a few weeks. The title of this one is Research Toward COVID-19 Antiviral Drugs. Unlike some of our past conversations, here you will hear about active research that could benefit tremendously from small donations. If you would like to donate to help with this research, please, you can text to give. If you text the number 41444, you text the letters PSCC to the number 41444, and you will receive instructions on how to give. Hopefully everyone can see the screen. I actually am not seeing a live screen right now. Is anyone else seeing a screen? Maybe we need to do a little extra work. Let's see. John is raising his hand. John, I'm letting you talk.
Okay, I took myself off mute. Yes, I can see you. I can see everyone.
Great, thanks, John. Are you able to see a screen that has the text to give number on it? I think that's the thing. There should be a
did not see that.
I just see you.
Okay.
I see you.
You see me too? Oh wow, I feel fortunate.
Okay, John, I'm going to remute you. Thanks a lot. We got one of our
thanks all
friends of the school, longtime friends of the school, John, who's and now I have maybe let's see. Okay, I think we're back. Well, we'll figure out that screen thing later, but I think we should continue.
You can also you can also see the text to donate in the Q&A session, in the Q&A area.
Great. If you go to the Q&A area, you can see that number. So let me continue. So as we'll go along and you're here in this conversation, you can utilize the Q&A button at any time during the conversation to ask a question. And I will be moderating and occasionally jump in and we can use some of those questions to facilitate conversation. So that'll be a way that everybody can join in. So let me please move on to the panelists. We're fortunate to have Dr. Rachel Martin. She's a professor in the Department of Chemistry and also has an appointment in the Department of Molecular Biology and Biochemistry. She holds a PhD in physics from, she holds a PhD in physical chemistry from Yale University.
Close.
Professor Martin studies the structure and function of proteins using a variety of biophysical techniques. She's currently working on understanding the function of key enzymes from the SARS-CoV-2 virus in order to facilitate the development of antiviral drugs. We also have her colleague, Dr. James Nowick. He's a professor of chemistry and a professor of pharmaceutical sciences. He holds a PhD in organic chemistry from MIT. His areas of research include bioorganic chemistry and chemical biology, molecular recognition, supramolecular chemistry, and peptide and protein structure. And finally, last but certainly not least, we have Dr. Chelsea Anorma. She is actually an alum of our chemistry department, and she holds a PhD in chemistry from the University of Illinois. She's now a postdoctoral researcher in Rachel's lab. In the Martin lab, she focuses on expressing and purifying SARS-CoV-2 protease. Protease for inhibitor testing. We'll learn a lot more about that today, and investigates properties of islands, crystallines. So with that brief introduction, what I'd like to do is I'd like to ask each of our panelists in their own words to tell us a little bit about how you got to this moment in your career, and then what you were doing when COVID-19 hit and how that may have shifted things for you. So Rachel, why don't you start us off?
Sure. Yeah, so this was not something that I was planning on working on, certainly at the beginning of this year. But when SARS-CoV-2 hit, I was teaching chemical biology, which is it's a big undergrad class. It usually has about 125 students in it. And, that was during winter quarter. So the class ended kind of mid-March when we first started to, to shut everything down temporarily. And so for the last lecture of the class, I actually talked about the virus and, how various key proteins work and, how it infects cells. And as I was getting that together, I started thinking about it. I realized that, we have the virus has this main protease that cuts up all its proteins after they're expressed. And it's actually related to some proteases that my lab has worked on before. So I started thinking about, what we could do to, to do something to help solve this problem. And that was on a Friday. And by that, Tuesday, we kind of had a project going. And I'll let James tell you about how he got involved.
So my story on this project actually begins more than 25 years ago with the AIDS epidemic. And when the HIV protease inhibitors were being developed, the life-saving drugs that transformed AIDS from a fatal illness to a manageable chronic illness, I became fascinated with the scientific process involved in developing these drugs. And at the time, my laboratory didn't have the skills or the technology to be making a contribution in that area. And there were many, many people who were, well more equipped and skilled to make contributions. But I was so excited about that work that I developed a molecular modeling exercise, a computer-based exercise showing the drug designs process to educate students who I was teaching in my molecular modeling class. When COVID-19 hit, we were getting ready to shut down the lab, and Rachel came to me and said, well, keep the deionized water running because we're going to need pure water in my laboratory to work on COVID-19. I'm going to keep my laboratory open. And at that point, the thought began to strike me, wait a second, we can do something too. If nothing else, we have the technology to assess protease inhibitors of the SARS-CoV-2 main protease that Rachel is working on. After about another week, I started to revive the old exercise that I had developed on HIV protease inhibitors and teach my own students who are sheltering at home working on this, teach them about that. And a very talented project scientist in my laboratory working with me adapted that exercise to invent a completely new molecule as a candidate against the SARS-CoV-2 main protease, the enzyme that's essential for the SARS replication process, the COVID-19 pathogen replication process, in a fashion that's analogous, but with very different details from the HIV replication. And the hypothesis that Adam and I came up with was so compelling, we felt we had to try it. So we applied to keep our own laboratory open. We had already applied and we filled out the paperwork, and Dean Bullock very kindly approved my lab's return, and we began working on this. So that's how we got involved.
I'll just jump to the punchline here. You have been initially quite successful in scoring a first hit on developing this molecule and finding something that might be useful in the future for really helping things along. And we'll hear more about that later. But Chelsea, let me ask you to tell us a little bit about your own story. I mean, we're always thrilled to have our own alums back. Tell us about that. Did you envision yourself working on something like this? And then, if you wouldn't mind, take a little time and explain to us, explain to a poor old astronomer here what this protease is doing and why this is able to fight this virus.
Right, sure. So as Dean Bullock mentioned, I'm actually an alum from UC Irvine's Department of Chemistry. I actually did my undergrad research in Professor Martin's lab, so this is a very familiar department to me. So UCI's chemistry department is really where I really fell in love with research and just like all the possibilities there are, especially in chemical biology, for being able to manipulate, molecules and use them to help, for human diseases. And so from that jumping off point, I went off to grad school in the University of Illinois, Urbana-Champaign, and I did my doctorate research there on mostly about cancer stem cells and developing molecules that can be used to detect them. So while it's not, enzyme inhibitors, it's a very kind of similar process, because I was looking at an enzyme that's upregulated in cancer stem cells and basically helping to test out a molecule that acts as a substrate for that enzyme and then basically glows, allowing us to locate that cancer stem cell. So it was a bit different, but sort of kind of similar mechanics. I was also working on an enzyme and molecules that fit into that enzyme. And so what when COVID-19 hit, basically this year, I was actually wrapping up my work there, writing up my thesis. I was actually one of the last few students in my department who actually had a in-person thesis defense, because right after that, everything started to close down. And so I was already in this sort of transition point of thinking where I would go next. And the opportunity came up to work in Professor Martin's lab on these proteases. So going back to Bolt's question, I was really excited to kind of jump in because these proteases from SARS-CoV-2 are really crucial for how the virus in general works. So the SARS-CoV-2 virus, basically, it has the genetic material to produce all its own proteins, the proteins that build up the virus. But they all sort of come out in one long strand. And in order to break that strand of proteins up into the individual proteins that are active and can build the virus, you need a protease, an enzyme that can basically cut up that long strand and produce the active protein. So because those proteases are so crucial for developing an active virus, a lot of labs have identified it, including ours. Our lab has invited as a key a key target for potential COVID-19 drugs. So that's why I'm very excited to be on this project as well.
Wonderful, that's fantastic. And Rachel, maybe tell us now a little bit about how the work that James talked about sort of will interface with what you're trying to do in your lab and this push towards ultimately developing treatment.
Yeah, so in my lab, we, as Chelsea mentioned, we work on proteins, excuse me, and mostly understanding their biophysical properties and mechanism and, how they do their thing. And so in the case of the SARS-CoV-2 main protease, of course, the main thing that we're interested about is its enzymatic activity. And one direction that we're going with that is looking at the impacts of mutation. So James mentioned being inspired by HIV protease inhibitors and how that really turned this fatal disease into a manageable one. And one thing that has been a problem with that process, or that has made it more difficult, is the impact of mutation, because viruses are mutating all the time. Now, we're lucky that SARS-CoV-2 does not mutate as quickly as HIV, kind of per passage through a human, but it still is mutating all the time. So the first paper from my lab on this is about the mutations and what kinds of substitutions are happening. And so that's actually been quite an undertaking. We have an undergrad and a grad student who are tracking every mutation in the main protease every day. They're collecting the genome sequence of every variant of the virus that's being sequenced worldwide. And then they've written a bunch of scripts that take those main protease variants and run it through a pipeline where we can do molecular dynamics simulations and predict what those proteins are going to look like. And this part is being done in collaboration with Carter Butts and his group. And so our first paper on this is describing, what the first, I think we had 79 mutations. This is as of about April 21st, what these things look like, so are there, and it turns out that there are trends in what kinds of amino acid substitutions we see. And at this point, the mutations are, they're due to what we call neutral evolution. It means that they're not under any particular pressure externally. Once we start putting in protease inhibitors, then they will be under pressure and, we'll start mutating against the inhibitors. And so the goal of this kind of research is that we're interested in mapping out the mutational landscape. We want to try to understand which residues in that protein can be mutated without negatively impacting the function. And maybe that will help us design inhibitors that are, more robust to mutation. We can start building that in right from the outset. So instead of, as James described his starting point, building in these models, for the wild type protein, as a next step, we can also do that with some of the mutant proteins that we know are active and try to come up with, next generation inhibitors that will actually be resistant even as the protein continues to mutate. So that's kind of the goal of what we're doing. And so we're currently working on, making a lot of the protein for, first we need to make a lot of it in order to have enough for all the various types of testing that we need to do. And then we're going to need to make a bunch of the mutants. We can't make them all. That's not realistic. I mean, even 79 is a lot. But at this point, the last time I looked, we had more than 250 mutations of MPRO. And so we're working on the next paper, the next theory paper on that. But so that's kind of the direction that we're going. We want to be able to design mutation-resistant inhibitors from the beginning.
Yeah, that's fascinating because I guess sort of big picture here, the worry is the virus is mutating and changing, and so you build a technique that inhibits one version of the virus, it just mutates out of it and keeps burning through.
That's right. So since we're able to look at, we have all the genome sequences of this virus since it emerged, which is a really amazing opportunity because we can look at, in this period when it's not subject to protease inhibitors, we can look at kind of what are the mutations that it can tolerate. And, at this point, we're not seeing a lot of evidence that there are particular strains of the main protease that work way better than the original one. That's not true for the spike protein. People are seeing mutations of the spike protein that seem to confer a big advantage. For the main protease, we haven't seen that yet. But at least we have an idea of what kinds of mutations can be tolerated. And so we can kind of anticipate where it's going to go if it starts getting pressure from these inhibitors. And hopefully we can design around that.
That's fascinating. James, can you explain to us then, what were you using to build your initial molecule that seems like it might be promising? And then how did that compare ultimately with what you'd like to do in collaboration with Rachel?
So what we were using to build the initial model, and this was a stroke of genius from Dr. Kreutzer, from Adam Kreutzer in my lab. Most people were starting with crystal structures of the COVID-19, the SARS-CoV-2 main protease, but SARS came along way earlier, about 2003, and is almost identical. And there was another structure that Adam looked at, which had the tail of a peptide, the tail of a substrate bound to the protease. And he got an idea that it could be connected into a ring. And I suggested the, shall we say, golden spike to close that ring. And so we were using computer-based modeling of an X-ray crystallographic structure, an actual structure where all the atoms are in the protease enzyme and where all the atoms are in a substrate molecule that fits into it, to design a surrogate that fits into it in a similar way. So sort of an enzyme is a lock, and we had a structure of the key in the lock, and we set out to design a fake key that would clog up the lock that would look similar enough to have the right grooves to fit into the lock, but wouldn't turn the lock and would gum up the work so that the virus couldn't use its enzyme to reproduce.
And what were you using to test it on if you didn't have it?
Okay. So,
Yeah, that's
So-
cool.
so the testing was done in an instrument called a plate reader with a substrate that lights up, that fluoresces when the enzyme cuts it. Now, the sticking point was the enzyme, and thus far we haven't. Rachel is working really hard, and Chelsea are working really hard as well. Rachel and Chelsea are working really hard to make lots of the protease. Right now, we've been buying minuscule amounts at exorbitant prices, spending all of the money that we can scrounge up to buy this to do these tests. So ultimately, when Rachel and Chelsea are successful, we'll be able to accelerate the research and run these tests on our molecules much faster and more efficiently, and also generate our own X-ray crystallographic structure of how the molecule that we invented fits into the protease, so we can make even better molecules that fit in even better. And that's what we need to do right now.
So I want to point out one more connection here, which is kind of neat. So the details of how to do all of that, with the inhibitor definitely came from Adam, but one of the specifics about what to do with it came out of a conversation that he and I had. And that was the discussion of how a lot of times in agriculture, people use the pro sequences of an enzyme in order to inhibit that enzyme. And so the way that works is if you have a protease, it's an enzyme that cuts up other proteins. You can't just express that and let it loose in the cell, right? Because it would go around and cut up all the proteins. And so often they're expressed with a pro sequence that blocks the active site, and then it gets cut off at some point later when it's ready to use. And so in agriculture, people make plant proteins that help the plants be resistant to pests, and then use the pro sequences to actually inhibit that enzyme. And I've never seen anybody do that in antiviral research. And so I had a discussion with Adam about this. And so that was why he decided to start with the C-terminal pro sequence of MPro to design his inhibitor. So it's a really great collaboration, and we're really able to bring together a lot of ideas from really disparate areas to make it work.
Great.
Okay,
Great
let me
point,
just jump
Rachel.
in. I have to jump in there and sort of emphasize that level of sort of transdisciplinary collaboration is atypical. And it's one of the things we're extremely proud of at UCI, that kind of interaction happens. And mind you, this happened while everybody was physically distanced. So he must have done this on some kind of like Zoom call. I don't know how it happened.
Slack.
But,
We have a Slack channel that all the labs involved in this are in, and we send stuff back and forth.
I love it. I mean, this is just wonderful. Chelsea, kind of jump in here for me. We talked about Timeframe, and James talked about, boy, you can't wait till you guys can produce what he needs at a fraction of the cost and even better. What's the time frame from your point of view? How soon will you be able to sort of start providing these protease?
So yeah, time frame is one of the questions that is hardest to answer, I think, because usually, like, you have an idea in your head, and usually when you go into the lab, there's so many new things that pop up that you even weren't aware of. So it's tough. We've been like a few weeks away, or like a few days away several times, and realize, oh no, we have to go back and check something else. So it's a little bit tricky
so
to
hard.
answer.
Yeah, in addition to Chelsea, Marquise Crosby and Mark Sprague-Piercy are working really hard on this. And this is a great team. They're trying a million things in parallel. And we meet about this twice a week, and we're almost there. We can make a little bit of the protein, but not enough to do what needs to be done yet. We're getting there though.
Talking about time frame. Let's take a step back here, just sort of for context. I think a lot of people are hearing about a vaccine that, you know, if all things go well, we might start having in the beginning of the year. How do new treatments like this sort of fit within the landscape of how we expect to live with this virus over the next years and into the future? What role will treatments like this play? I don't know, who wants to take that?
So I think that treatments like this have an important role even once we get a vaccine. Because for one thing, there are always going to be people who are immunocompromised and are unable to get the vaccine. And also, we just need it in the arsenal for the next time. And in particular, it's not even so much that we need the particular molecule that, you know, that we end up generating. We need the toolbox. We need to be able to boot this up quickly and do it every time there's a disease like this that emerges. And in general, you know, I think, you know, of course, the vaccine people are really improving the speed of their pipeline too. But in general, I think a lot of stuff like this, you know, could be faster to come out than a vaccine. So it's also an important piece of the puzzle.
One of the sad things is, had this work been done back when SARS came out back in 2003, the drugs that would have been able to treat SARS would have been almost perfect to treat COVID-19. The viruses are so similar that a drug that inhibits the protease would have been good enough. So right now what's happening is people are scrounging and trying to find antiviral drugs that are out there from other areas that are not closely enough related. So these are all what are called coronaviruses, and SARS and COVID-19, SARS-CoV-2, as well as MERS, and they're all pretty closely related. And if we look at the timeline, sadly, it's unlikely that this is going to be the last big epidemic from coronavirus. So the work that we do now not only will hopefully provide tools for people who can't or won't take the drug, either as can't or won't take vaccines, either as treatment or even as preventative, as pre-exposure prophylaxis, it's called. In other words, a treatment, a pill you could take every day to prevent you from getting sick. But the other thing that can come is to have the tools in our toolbox to fight the next coronavirus epidemic that might come in the future.
So Chelsea, as the you know, postdoc, young scientist with your career ahead of you in the group here, not that Rachel and James don't have tremendous success in the future, to put it that way. I gotta watch myself here.
Yeah, we're the old scientists. That's okay. So.
You know, how do you see the landscape? I mean, do you think this may change the direction of your scientific career in a little bit? I mean, are you at a point where you're, you know, where do you see your future and sort of scientific interest headed, given what you experienced for the last few months?
Well, I think kind of going back to what James was mentioning, I think a lot of scientists or just people in general are realizing how important it is to learn from, you know, the things that are coming up and really put that emphasis on developing the science as much as possible. And also, I want to say I see a lot of emphasis on more collaboration. Like UC Irvine obviously has this wonderful department that, you know, ever since I was here back for my undergrad, I've always marveled at how open and friendly everyone in the department is with each other. It's definitely something I contrast a lot with, you know, other departments that I've seen. But now, yeah, not only UC Irvine, but so many other departments across the country, I think, are working together a lot more. And I think that is definitely a trend that will continue. And I really excited to see what comes out from that, because so often, you know, just that little communication between the labs or departments really can help you make that boost forward that would take you much longer otherwise.
I'll add that since starting this, we've made a connection with another scientist who's working with a mouse model of coronaviruses and now is gearing up to work with SARS-CoV-2. And so we will, I think, in short order, be able to have a situation where we will be able to test our molecules and other people will be able to test theirs in animal models of the disease, which is going to be very important in advancing this. Based on what we've done, we were contacted by another scientist, a UCI person who's currently in Norway, who's doing X-ray crystallography, who can rapidly determine the structures, the three-dimensional pictures of the molecules in the protein to see how they fit. So yeah, science is fantastically collaborative and UCI is the most incredible environment to be working collaboratively. Picking up on the theme that Rachel mentioned earlier about her conversation with Adam, this is something that is so integral to doing science. It's the random conversations where you don't necessarily go in and say, let's meet and plan something. Mm-hmm. You're gathered around together and you're talking. And that's also one of the hardest things about doing science during the pandemic is that we physically can't gather together around an instrument or around a data result and look at it. We have to be apart or remote.
Be real, James. These conversations happen at the coffee stand. Everybody knows it.
True.
Well, I will comment that I'm strongly advocating that there be a pretty robust coffee stand in this building once it gets going, because we know that that's where the magic might happen. So we'll hope that happens as well. Let me sort of shift. We're getting a lot of interesting questions in the Q&A box, and I want to sort of bring in a few. One of the questions that's come up is safety. You know, we hear a lot about needing lots of, you know, phase 3 testing for the vaccine. When this, you know, you're successful, you begin to sort of do tests for this methodology. What would that look like? And do you expect that there will be sort of potential negative impacts, or what's your read there in the longer run?
I guess I'll comment. So UCI has fantastic research facilities and so my laboratory is not working with the virus itself. We're working with biochemicals that themselves are harmless. So the biggest danger in my laboratory right now is other scientists, in the sense that we are all having to act as if we might have the infection or our peers might have the infection. So that's currently the biggest safety danger in any of the research laboratories here. Now, UCI has fantastic facilities, and we have biosafety level 3 research space, BSL-3 space, that is suitable for working with the virus. And the collaborator I mentioned, biological collaborator, has received approval to work with the virus and to use that space. So his laboratory personnel will be very well protected. And doing that research, Rachel and I work at what are called BSL-2 conditions, and that just involves use of a cabinet and use of eye protection and a lab coat, whereas BSL-3 conditions involve being much more heavily covered, much better control of ventilation and access and face coverings, you know, not just face coverings to protect you from transmitting it. And so his personnel or other personnel who engage with the actual virus will have that higher level of protection.
Yeah, that's interesting. I mean, the sort of a view into what it's going to take as we move forward. So once we have an inhibitor, what is the expectation for the safety of a drug like this? Are we worried that we're going to do lots of trials, or inhibitors tend to be somewhat safer than other kinds of drugs? I have no idea, just asking broadly.
It definitely will need to be tested, and we'll need clinical collaborators for that when it gets to that point. This one has a good, it's a good scenario because there aren't any human proteases that have the same cleavage site as the SARS-CoV-2 main protease. So, you know, the things that are, you can rule out the things that are just toxic because, you know, James and his team can test that in cells.
Mm-hmm.
And things that might have off-target effects to human proteases that are really important are going to be less of an issue here just because the substrate specificity is so different from any of the human enzymes. So I think this one is better than, you know, than it might be in terms of those issues.
Well,
So there's a very long process to advance a molecule from an initial hit to a drug, which is what we have now, which is just a first step, to a drug candidate that has more promising properties. And then through preclinical work that involves testing for various types of toxicity in animals. So you can give and treat a mouse with the molecule and see if there are adverse effects on the liver and the kidneys and other parts of the mouse. You can study whether the molecule is broken down rapidly or slowly in the animal's bloodstream. And then one can move to actual animal studies for treatment with the drug candidate, where you have an animal model, whether it's a hamster model or a related model, and eventually a monkey model before it ever gets to human beings.
We have a question here from our director of scientific computing, and it has to do with computing, shockingly. The question from Domingos is, you know, imagine this would have happened 15 years ago. How much harder would it have been for you to sort of make the progress that you have without the computational facilities that we have and other technical advances that have been made, you know, in the last decade or so?
So the computational stuff, we couldn't have done it. We couldn't have done any of it. So for one thing, you wouldn't be able to have genome sequencing of every single SARS-CoV-2 infection that, you know, that has been recorded all over the world in the way that we do, because, you know, just sequencing those genomes is computationally intensive. And then also doing that large scale molecular modeling absolutely depends on having GPU technology. And so we just wouldn't be able to do any of that if it had happened 15 years ago.
The other thing that's been transformative is data transfer. So some of the data sets that are processed, particularly in X-ray crystallography, are very, very large. And having the bandwidth to transmit and process those data sets is also critical. Even in the molecular modeling area that my own lab is doing now with just our own computers, that used to require highly specialized computers. So remember, I talked about developing an HIV protease inhibitor exercise 25 years ago. We used to have a special room with special silicon graphics computers that at the time seemed like such a tremendous resource and so valuable. And the computing technology has progressed so rapidly that those computers are now doorstops or landfill or recycled. And now even a desktop computer can make advances on simple problems. And as Rachel points out, a cluster and GPUs for the type of number crunching that she's doing can be incredibly valuable. And UCI is really well equipped.
And I have to say that you know, that the thing that has really driven that technology is gaming. I mean, I like games, but I mean, we really owe them a lot because that's what has pushed forward the GPU technology that we're using.
Yes. Chelsea, I wonder, you know, we have a few other questions in the chat, so I'm just going to throw one at you. I'm interested in your perspective. Do you know of groups outside of UCI that are looking at similar, you know, approaches is them to collaborate with folks or learn from folks who aren't UCI?
Yeah, definitely there are a lot of labs that are interested in this. I wouldn't know a lot of the specific details on different groups who are doing this exact type of inhibitor design, but we're definitely reading about different efforts all the time. And yeah, if ever there's something that we need to collaborate on, I'm sure we could definitely reach out. Um, yeah, I don't know of any specific group though that I could off the top of my head.
Yeah, I'm in contact with a lot of the groups around the world that are working on this, and a lot of it is, some of it is direct collaboration, but a lot of it is just keeping in contact so that we don't reinvent the wheel. We don't want to all work on the same thing because it's kind of a waste of resources. So James mentioned Hudel Luca in Norway. We're in contact with him. We're going to collaborate with him. There are also some groups in Germany and Switzerland and China that I'm in contact with. Mostly it's just like, hey, here's what we're doing. What are you up to? Just to make sure that we kind of stay out of each other's way and don't duplicate efforts.
So let me shift the question and ask more broadly. To each of you, what worries you most about the current crisis? What concerns you the most, and what are you focused from your point of view?
Sorry, go ahead, Chelsea.
Oh, go ahead. Well, I just was going to say what probably concerned me most, honestly, is misinformation, honestly, and just kind of the public not really understanding where the science is, and so basically getting led astray by folks who sound sciencey, but don't actually, have who aren't actually active in this research. So I think definitely sort of that public education would be very important.
Mm-hmm.
Yeah, that's a really good point. I was going to say something similar, just that in the public discourse about this crisis, science is not as prominent as I think it should be. There's a lot of arguments about should we close things or not, what should we keep open, should people wear masks, all of that, and all of that is very important. But I think that science has not been emphasized as much as it should be, because the goal is not to close everything down forever. We can't do that. That's ridiculous. The goal is to, buy time to solve these problems. And that means that as a society, we have to invest in that. And, we really need the public to be more on board with, what it's going to take and, what sorts of things are happening.
I would agree completely. And this is a really scary time in terms of misinformation on everything from vaccines to junk cures or treatments for diseases. And the fact that people are afraid to really, go ahead and listen to expertise gained through experiment and knowledge. And are sort of pursuing things that don't make sense, whether it's not wearing a mask or not vaccinating one's children against regular childhood diseases, or taking drugs or supplements that are bad for you, or assuming that they provide protection.
Yeah, I mean, I imagine it must be frustrating to each of you who's sort of spending many of your waking hours trying to do things the right way and develop something that actually works. And then there's sort of a snake oil being sold left and right. I can only imagine how frustrating that must be to witness. In terms of other treatments that potentially could work, are there other things that are going on that you're aware of that you're interested in? That's one question. Other general approaches that you might be aware of. And then another question that's come up in the chat is there a role for CRISPR-Cas9 in sort of developing treatments for this virus?
Well, other treatments. So one of the very exciting therapeutic approaches, so non-vaccine approaches, is to prevent the virus from getting into the cells that get infected in the first place. And this is an approach that many people have taken. They've also taken a similar approach in AIDS, by the way, although that's not as prevalent, as widely used in the arsenal of drugs to fight AIDS. There's even a drug called Fusion that prevents the HIV virus from infecting cells. And so I think there is potential promise of drugs to prevent the virus particles from getting into the cells and infecting them.
Another good one is the drugs that inhibit the replication of the RNA. So there's an RNA polymerase that replicates the genome, and some of the drugs against that look really promising as well. And that is also a strategy that's been used with HIV. So some of the very earliest HIV drugs were of that type. So these are all, you know, other things that people are doing that, you know, that are good strategies also.
And nowadays,
Yeah.
many times you need a drug cocktail. You need to treat diseases with more than one drug in order to suppress it, where you use two together and you get actions that are more effective than either of them alone, the whole being more than the sum of the parts.
That also helps fight evolution against the drugs, because if you're hitting it through two different pathways, it's much more difficult for the virus to evolve around the, you know, the drug.
Chelsea, were you going to add something? I want to make sure you're heard if you have a...
No, James and Rachel cut me off at the pass, so basically I was going to
Sorry.
agree that that yeah, drug cocktails I think would definitely be a big, you know, something really important in the future for sure.
We expect this virus to be with us in perpetuity. I mean, I know that no one can read the future, but just given your knowledge and this, do we expect this to sort of be around even if we have a vaccine and it won't be 100% effective, et cetera? What do we think the next decade's going to be like?
I think nobody knows, but one thing that is for sure is that things like this will be with us. You know, even if this one gets completely eradicated, which I hope it does, there will be new coronaviruses that emerge, and I think that's just clear from the past that, you know, that they have come up every once in a while. And I think what we should learn from this is that, you know, we need to do better at tracking these things as they emerge, and having an ongoing program to, you know, to fight them and have the tools to develop drugs as these things happen. Because as James said, if somebody had done this in 2003, you know, we would have a drug that would probably be good enough. And, you know, people were working on it, but then as soon as the threat passed, they kind of got defunded and moved on to other things, which is a shame.
Yeah, I would agree with what Rachel says on this. It's really hard to tell. You know, many things go away. Year's flu virus goes away, although a new strain will emerge for the next year. But, you know, there are very few viral diseases that have been completely eradicated. And now we see with resurgence of measles that one always has to be vigilant. Even Ebola, which doesn't actually spread very well because it's too dangerous, it burns itself out, has kept popping up. So it's hard to tell whether this will be a chronic human problem. And of course, if there is a drug or a prevention, it may be possible to completely keep it under control. When I've traveled to areas where malaria is prevalent, I will take, and I hope to continue in the future to travel to India and other places. I'll take a malaria preventative medicine so I don't get it. So who knows?
Yeah, I definitely agree that whether or not this particular coronavirus is around, there's always new things coming up that we need to be prepared for, especially in sort of today's society where, you know, travel between countries is so rapid and easy. Unfortunately, that also means that, you know, diseases that used to be localized in one area can now be quickly spread throughout the world. So definitely need to develop those tools so we can be ready when that happens.
With globalization or with growth and destruction of habitats as well, we see more incursions into places where there may be an animal disease that can then jump to humans. And with global climate change, with the warming of the environment, we see more rapid development of diseases as well and more destruction of habitats. So yeah, it's going to be all the more important to keep working against this and other diseases, not just coronaviruses, in the future.
Can you, let me ask a couple more questions. We've just got a few minutes left, and I'm going to take prerogative and ask my own here. James, you're former department chair, Department of Chemistry. So I'm going to ask you to speak on behalf of your field. What is the role of a chemist? What is the role of chemists in our fight against viruses like COVID-19 and other disease, other viruses we expect in the future?
What many people don't realize is the invention of new medicines starts with chemists. So the biggest sub-area of the Department of Chemistry is organic chemistry, broadly defined. It's the area that has the most PhDs given in the department. And one of the reasons there are so many people there is many of our PhD, and for that matter, BS students bachelor's students from the department get hired by the pharma and biotech industry. So there's just a bigger pipeline of people coming through. And one of the big jobs in industry for organic chemists is called a medicinal chemist, as involved in either discovering new drugs or in coming up with ways to make them. That's often called a process chemist. So chemistry literally is the central science in the invention of drugs and medicines to fight not just viruses and bacteria, but also diseases like cancer and heart disease.
And physical chemists develop the instruments that we need to do to study those things and understand how they work.
Indeed, and we are very much hoping to be able to use NMR spectroscopy to understand how our molecules interact with the protease enzyme.
And I guess
Thanks.
chemical biologists just dabble in everything and put everything together.
Yeah, we're all working together, but this is it's interesting to think about things. So what's in our last 5 minutes here, what's next? What are you hoping to accomplish in the next month or two? Chelsea, why don't you jump in first?
Well, my number one goal obviously is to get Mpro expressed. So like I said, we've been really close several times, and we've got 3 of us on this team, as Rachel mentioned, who are really trying out a lot of different approaches. So we're really hoping that, one of those has got a hit, and we'll have the main protease ready to go for testing. And then, going from there, testing out the inhibitors and, starting along those first steps towards seeing how well this works.
Yeah, definitely. Yeah, as Chelsea said, definitely scale up. So being able to make large quantities of Mpro so that we can test all the inhibitors that we want to. And of course, we have other collaborators in addition to James who are working on different molecules. And so we need to test those too. And also, one of the things that I'm really excited about is being able to, once we're able to make larger quantities of the native protein, then we'll be able to start testing out some of our hypotheses about the role of mutation. Because we can make, once we have the protein expression going very well, we can make point mutations very quickly. And so we can start looking at how those things actually work. And that's when we really start learning a lot about the mechanics of how the enzyme does its thing.
So within the next month or so, I hope to have a X-ray crystallographic structure of how to find out how well the molecule UCI1 is fitting into the enzyme, and to use that X-ray crystallographic structure to design new homologs, new related molecules that fit even better and work even better. So that's one thing. People in my laboratory, Adam and others, are also busily designing yet other molecules to fit into the enzyme. So we have another candidate that I can't wait to test out that's related but different in structure. So we want to keep inventing molecules and coming up with molecules that work really well at blocking the enzyme, getting into the cells where they're going to need to act to protect the cell from the infection or to prevent the virus's replication, and then eventually to start to look at how they interact with either the virus or the related virus I mentioned, the mouse coronavirus.
So how does that work? You know, you've got UCI 2, 3, 4, and when you land on 5, that's the one that you're very excited about and you think will be great. You're making this up. What's the next step for testing? Are there collaborators at UCI that you would work with, test this on animals? You know, how does that progress?
So that would be one step. Let's say UCI 5 is the one that has high activity, no toxicity, which UCI 1 does, gets into cells and blocks within a cell model the replication of the virus. So at that point, next we want to get into an actual, the replication of a model virus. At that point, we want to get into an animal model and see if it can prevent the disease from propagating in an animal, like a mouse or a hamster. If we are able to raise the resources, we can currently want to check how the molecule is handled within the bloodstream of an animal, whether it is broken down quickly or slowly, whether it has adverse effects in the bloodstream. And so this is called preclinical work. And often that can be done even more efficiently by a company, so that we're looking at companies, or we have the capabilities to do that here. UCI has fantastic laboratories, and one of the key laboratories for that would be our mass spectrometry laboratory that would be able to basically do the analytical studies needed to help determine these properties of the drug candidate.
Fascinating. Well, let's hope that begins to happen. I mean, that's what we're all hoping for. Rachel and Chelsea, do you have any last words? We've got about a minute left. What should we be closing with? What are you thinking about these days?
Chelsea?
Oh gosh, too much pressure. I froze. But basically, no, just keep your local scientists in mind. We're working really hard, and yeah, send us some good wishes, I guess, so we can figure this all out, so we can get back to some kind of normal.
Yeah. Okay. Well, Rachel, you know, thank you so much for spearheading this from the beginning. We're so proud of all the advances that have happened already. We're proud of our alums like Chelsea, and we're hoping for the best here as we move forward. I'd like to thank everybody for joining us today for our 4th installment of Compelling Conversations. I hope you've learned a lot, and we're going to have our 5th installment very soon, so keep a lookout for that. This will also be posted on our YouTube channel and I think our homepage as well. So we'll send that out. Thanks very much. Bye-bye.